All right, so I have some more specific and detailed lectures on several of these subtopics, but this is kind of a bigger overview of everything we get into with cells, energy transfer, energy storage, metabolism, and a lot of the key terms that are related to those. So this is really the place to start looking at that. So what happens inside cells is a lot of transfer of energy. So energy being transferred and transformed from one form to another form. So there's some basic terminology and some concepts for you to get around first. All energy for pretty much all living things on Earth comes from the sun. And although you might say, well, you're not photosynthetic, you're not a plant, so your energy doesn't come from the sun, except that your energy comes from your food. And your food is either a plant or an animal that ate a plant. And so all the energy from the sun gets transformed into organic energy and organic molecules and bonds within those molecules through the process of photosynthesis, which we will cover at some point later in the course. So when we talk about energy, going back to really super basic traditional terms, there's kinetic and potential energy. So potential energy is what we're going to be focused on mostly in the class, and all the different forms of potential energy. So potential energy is going to be stored in chemical bonds. So when you have a bond, which we've already gone through, between elements, there's energy in that bond. It costs energy typically to build it and stores energy in it until it's broken, and when it breaks, it releases energy. Right? And so some of those concepts around energy have to do with what are referred to as laws of thermodynamics, which seems out of place in, say, a biology class, but it does relate directly to this whole idea of metabolism and energy and enzymes and things that we are going to cover in the class. So, laws of thermodynamics. And it's specifically related to something in this class that people, and even in the higher level majors class, that people always get wrong or misstate, and it's really very incorrect, right? To think certain things or state certain things related to the laws of thermodynamics. So the first thing is energy, in whatever form it is, cannot be created nor can energy be destroyed. So you can't create energy, you can't destroy energy, right? So cells don't create energy. There's nothing, mitochondria don't create energy. Nothing creates energy, right, in cells and living things. Okay, it's basically taking energy from one form, breaking a bond, releasing then that energy, and that energy is then used to do something else. It's stored some way, it's transferred or transformed. Okay, so energy. Okay. Why? Okay. Is transformed. And so what we're going to find are things such as, stuff we'll get into in this block, say related to membranes, right, which we've talked about now a little bit already. So you could have a protein, right, in a membrane. It might require the use of ATP. Right? The ATP gets broken into ADP and phosphate, and as it's broken, that gives off energy. That energy allows, so that energy is then transformed into this protein who can use it to do a job. Maybe it's pumping something else, so like a sodium ion, across the membrane from low to a high concentration. So over here, maybe it's sodium low, here's sodium high concentration. So that required energy to push it across. So the energy was transformed from a chemical bond holding together phosphates. So it was like phosphate, 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 triphosphate, right? 3. One of them was broken off, it became diphosphate. And as it was broken, that energy can be used to do this. Now it's not lost, okay? It's stored, it's transformed. Now the energy is in a concentration gradient. Sorry about the squeaking. I'm going to try to... This pen writes better than the others, but it squeaks a lot. So the others are all sloppy, but they don't squeak. So it's going to trade off. So the... now it's here. So some of it's going to be lost as heat. So the heat, it's going to be heat in the system, but heat's just going to be transferred to other things and transformed into other things. So there's going to be, some will go as heat and some will be completely stored, but now as a concentration gradient. So from bond to gradient, that sort of thing. Second part of thermodynamics, this is usually called the first law. And anywhere that you look this up, search first law of thermodynamics, you're going to find anywhere you look online, any book, something that sounds almost exactly like this or very similar to it. If you go and search up the second law of thermodynamics, you will find as many things as you look up, 10 places, you'll find 10 different statements. They may not even seem related to one another. Right? The second law of thermodynamics deals with a concept called entropy. Okay. What is entropy? Entropy is disorder. So entropy will increase often as energy is transferred. So as energy is transferred from one form to another, and sometimes transformed and from one way, very different way to another, often disorder increases in the universe. Another way of saying this is that nature, this is really the best way probably for you in this class to think about it, is that nature favors disorder, right? Or entropy. So a more natural state of things is to move towards states of disorder. So now think about concept again. We've already gone through diffusion, right? If I have molecules and I were to pump them into this space here, there's this whole big space, but for some reason I've divided it into two, and this side has a lot and this side doesn't. That means this is order. It's on one side, not the other, right? So what's going to want to happen is they're going to want to move to this side, right? By diffusion. Why? Why do they want to move to that side? They want to move to that side because this is now high energy, because they're smashing into each other because there's so many, and also because it's ordered. And so energy is going to then be released as it goes toward a state of more disorder, which means just spread out everywhere. So as opposed to a whole bunch of things in like one little spot, and a whole bunch of things in one little spot, and a whole bunch of things in one little spot, they're kind of clusters. Instead, you have disorder where there's maybe no particular pattern and things are just kind of scattered everywhere. So that's entropy versus order and putting things together. So while nature favors disorder, living things are going to constantly be using energy to try and create order. So building molecules makes them ordered, right? We start to order the atoms and the electrons together. That's going to require energy to do that. Creating concentration gradients, that's creating order, and that's going to require energy to do that. So there's going to be all these energy transfers that are happening, and that's a lot of what's happening in the cell. So the membrane proteins need energy, or they help energy become released. So there's always some kind of energy conduit. They're kind of channeling energy in one way or another. And now, inside the cells, inside the compartments of the cells, what we're going to have then are chemical reactions taking place. Okay. So reactions, I'm going to get into this in a second as soon as I erase this. Reactions are where you're going to start off sort of with one molecule and then it's going to change or be transformed into something else. And in some reactions, they can occur sort of on their own without any help from anything else. They'll just happen. because they're favorable, because they're going to create more entropy or more disorder. And there's other reactions that would then order things, and those would never, ever, ever happen on their own. But they do happen in cells because cells can then take energy from one thing and use it to do something else. So when we have reactions in cells, so we're going to talk about biochemical reactions. We'll have some molecules that are going to be the reactants. So usually one or more than one thing. And then we're going to get a product or multiple products. So for example, we have, A plus B equals a new molecule AB. So A is a molecule, B is a molecule. Now AB is a new molecule that's combining the two of them together. That would be a reaction, a biochemical reaction. Or we could do it the other way, where AB breaks down into the two separate. These are now separated. So one of these reactions would or could potentially just happen on its own. And one of them would never, ever, ever happen on its own. So this reaction here is a reaction that we refer to as being spontaneous. And the emphasis is can, not that it will. Okay, on its own. So spontaneous reactions can happen on their own, not that they will happen on their own, right? So what's the difference? It just means this is possible, and sometimes it may occur, and sometimes if it occurs, it probably isn't happening very fast. So you sometimes think of the word spontaneous as meaning quick, but not necessarily quick. It just means possible here. Okay. Versus this one up here, which is non-spontaneous, means it would never happen on its own. And it's kind of like the ball rolling up the hill, right? So if you have the hill like this, and we have a ball at the bottom of the hill, will it go to the top? Ever? No, it'll never ever go up the top. That's non-spontaneous energetically. Could it be pushed? Yes. So this type of thing can happen in cells. You can build things and combine them, but what's going on here? It requires, I'll use the other one so it's quieter, requires energy. Okay. And then you have the other version over here where you have, say, the ball at the precipice, right at the top of the hill, ready to go down. So can it go down the hill? Sure. If it starts rolling, it's definitely going to keep rolling downhill. Will it? I don't know. There's a lot of factors that might affect it. Maybe it's kind of stable where it is right now. It's not. There's a little tiny pebble blocking its path. Something, holding it back. So it's not happening at the this moment in time. But it could happen, and if factors were right, it would occur. And if it does occur, it's actually going to give off energy. So this type of process will release energy. Okay, so spontaneous reactions release energy, non-spontaneous reactions require energy in order to occur. Now, where enzymes fit into this, so the next thing is we're talking about enzymes. In cells, we have these proteins called enzymes. And we're going to need more space, so I have to erase this over again. And the enzymes are going to be the ones carrying out both kinds of reactions. So typically, while there are some reactions that can happen on their own, most reactions would happen on their own so slow that the cell wouldn't really be able to survive or be alive. Okay, so most reactions do not just happen all by themselves. Most reactions require the help of an enzyme, even if they are spontaneous. So even if there's something that's possible to happen all by themselves and will give off energy when they occur, they still usually won't happen. All right, and that's because of this. So this is the curve here that we're going to draw. So instead of the curve just being a downhill like I was drawing before, we're going to do something else. A real curve for most of these biochemical reactions, okay, looks something like this. So G here is going to stand for a person's name, okay, Gibbs, but it usually represents what we call free energy. So what's free energy? Free energy is going to be available energy to do work in a cell. So inside a cell, this energy that we could use to do work. Is it going to be used? Maybe not, and maybe not all of it, maybe some of it, but it's potential. So it's kind of like how much potential energy do we have? It's there, it's potentially available to do work. So this is kind of where we have our reactants up here. So this is like our AB. The idea is if it breaks apart and increases entropy, so if A plus B, where it breaks, that's going to release energy. And be a spontaneous reaction. But you see the hill here doesn't just slope down. For some reason here, I have drawn in with the hill a little bit of a hump here, an increase. So it's gone up before it goes down. So there's some kind of energy input required. So even though this is a spontaneous reaction, even though it's going to give off energy, it doesn't just happen all by itself. It needs some kind of help in order to occur. This input is called activation energy. Okay, so activation energy. Sometimes it's written as E, both capital, E and A, but the A is like small and the E is really big. So it means energy of activation or activation energy. It's the same thing. It's the amount of energy required to get a spontaneous reaction started. And a little note I'll throw in here, it's like striking a match. Okay. So how is it like striking a match? Think of a match. It has stored potential energy. It can burn. And when it burns, it's going to give off energy as light and heat. It's going to burn and burn and burn and release energy and release energy and release energy. And eventually it's going to be in a non-energetic charred state where all the energy's been released. Okay, so that would be spontaneous. The stored energy of the match, the released energy of the burnt stump, that's all that's left behind. Okay, and so all that energy that's released would be free energy that would be released. How long would it take for you to stare at a match before it starts to burn? Right? So like never, right? So it's never just going to burst into flames. It requires you to strike the match. Okay, so what's happening when you strike it? Friction is adding heat. You're adding heat to give off heat. Right? So that heat that's required, the striking of the match, is the activation energy. It's we need to activate the match to kind of get it going and get it started. And most all the chemical reactions in cells are like this, where they require a little bit of energy to get going. And how much that is can actually be highly variable. So it can be a lot. Actually, for some reactions, it could be very little. For other reactions, there's no set amount. Different reactions have different amounts of activation energy that they require. So some things with just a little bit of heat in the environment just then happen. And if there's less heat, if it's cooler, it doesn't really happen as much or as often. So that's why temperature can affect chemical reactions and speed things up, because it's energy that's available to overcome this. Keep this in mind as well, that energy, does striking a match take away how much light the match can give off? Or does it take away how much heat the match would output? If you had to strike it once and it didn't work, and you had to strike it a second time, third time, fourth time, fifth, and the fifth time it lit up, does that mean it gives 5 times less light or something like that? No. So you can have a lot of energy required to get it going, but it has no effect. That has no effect on the overall change. So this triangle is delta, the Greek letter delta. And so delta means change. All right, so our total change in free energy is going from here to there. And however much of this activation energy you require doesn't matter. Okay, it's not going to affect that. It's always going to be the same amount of energy released. And so that's the kind of the basics of all the chemical reactions that are happening in cells. Now, where do enzymes fit into this? So, what do enzymes actually do? Well, because many of these chemical reactions require this activation energy, and it could just come from random heat in the environment, but many times it needs some guidance or help. So an enzyme will come in. An enzyme is a protein. It has a unique three-dimensional shape, and it can change shape, and that's really the key to how it's going to work. Okay, so enzymes will change their shape as they work. All right, and that changing shape is going to allow them to interact with other molecules and force those molecules to change shape. So an enzyme is going to be three-dimensionally shaped, and let's say it's going to have like little pockets like this in it. And in those little pockets, say a molecule, might be able to attach. Let's say here, this is still I'll use that simplistic model, our A, B. So this is A and B, they're combined, they're connected, together. And this up here is the enzyme. So enzymes are going to be unique and very specific for their binding. They do not bind anything or things that are similar. They only bind something that they're very specific to interact with. So you have to be really specific to interact with a specific enzyme. So two things that are really close to one another in terms of their structure and shape, an enzyme will be able to recognize them, one versus the other, and typically one it will ignore and the other it will interact with, okay, because it's both three-dimensional shape, properties like charges, positive, negative charge, polar, non-polar, all types of chemical properties are available in these binding sites. And so the binding site of an enzyme, so these things here where the molecules will bind, that's called the active site. So the active site is the binding site where molecules will attach. And so at first people didn't really understand how enzymes worked. So what did they do to speed up chemical reactions? And they had an idea, a model called lock and key. And it explained a little bit of how enzymes were so specific. Like a key is very specific for a particular lock. And you could have the same car as someone else and have a key from that same brand that's a Honda, and it looks the same if you're looking at it. It seems the same thing, but they're not the same. They won't open up another vehicle or start another vehicle because they are different. They are uniquely different than one another. So what's going to happen here is that it's very specific binding, but yet the thing is, does your key change? When you pull a key out, is it different or altered? No, it doesn't. Nothing actually happens. It's like the key is the same and the vehicle is the same. Everything's really the same. It's just talking about how they bind. So the question is with enzymes is, what are they actually doing, like to help the reaction, besides just attaching to something? So if I had two things connected like this together, and I just said I could grab onto them, and then I can release them, how does holding on to them and then just releasing it going to help it break apart into two separate things? It's not gonna, it's exactly the same as it was when I first grabbed it and I just released it. It's the same, nothing's different. However, if I grabbed it and then bent or changed shape, these were really easy to break, but it becomes much easier to break if they put some stress on it. So the idea is that enzymes and the molecules they bind to, the reactants in the chemical reactions, do not attach in a static state, right? Not like a lock and a key. So enzymes are not static. Are not static. So they change shape. Okay. So what happens is when the enzyme and this new molecule, that we called it the reactant, and now I'm going to call it a substrate, It's the same term. So substrate and reactant are the same thing. It's just what we call the reactant when it interacts with an enzyme. So when the enzyme binds this other molecule, like AB, what's going to happen is both the enzyme and the reactant, or the substrate here, are going to change shape. So enzymes and their substrates change shape. Both change shape when they bind. When they do that, what's going to happen is that's going to put those molecules in a state where the bonds are like almost broken. It's not going to totally break them. It's going to put them where they're just about broken, but they're not totally broken yet. So the old bond is still kind of there, but the new bond, isn't really formed yet. So it's something sort of in between. So what they do is, instead of lock and key, the way enzymes work is through what's called induced fit. So induced fit replaces, just like you hear on the board, replaces lock and key. And induced fit creates what's called a transition state. And that's where old bonds are breaking and then new bonds are forming. Okay, they haven't formed. So the bonds have not been broken, new ones have not formed. It's sort of an in-between, a transition state. All right, it's a state in between two different forms. So that's what enzymes do. Enzymes attach to things, they change shape. The things that they attach to change shape. That puts them into an intermediate state, and that means that they're really ready to be transformed into something new, like breaking the bond here into making these two separate molecules. And that's kind of how the reactions work. So we're going to go a little bit more into a little bit on how enzymes can be sped up or slowed down, how cells can give enzymes feedback, how they can say like work faster or turn off, they can be turned on and off and all those sorts of things, a little bit more. But this should give you a big, broad understanding of the concepts and the terms related to energy, energetics in cells, the chemical reactions. This graph is important to kind of look at with the free energy, the original reactants, the products, the activation energy here, and then what enzymes actually do to lower the activation energy. So that's another thing just to add in here. What do enzymes actually do? So if you're asked, what does an enzyme do? It lowers activation energy. And then someone says, so what? What does that mean? Well, it means then it doesn't take much energy for the reaction to occur, which means it'll happen faster, or it speeds up a spontaneous reaction. So a reaction that would already happen on its own and give off energy, the enzyme lowers how much energy it takes to get it started. So it's kind of like adding something to that match, so that instead of even striking it, I might just tap it and it just bursts into flames. It's something that lowered that point where it would ignite. And so that's kind of what an enzyme does. It accelerates the reaction so that it happens potentially faster. The reaction itself is going to happen at the same speed, but it's more likely to happen if the enzyme is helping it out. That's the key. All right, so there's a bunch of terms in here, and there'll also be terms that you can read about as well. And then we're not done with enzymes. I'm going to have another enzyme lecture that goes more into regulation and control.